it working now? Okay, yeah. Okay, I'll go over that again for everyone on Zoom. So for the background for nuclear receptor signaling, you have the ligand, which is a lipophilic greasy molecule, which likes to go through the cytoplasm so it can bind in the nucleus. Now it'll bind with the receptor in the nucleus, and that ligand binding creates a conformational change so the DNA can interact with it better. Now, these are some examples of some nuclear receptors. They should look familiar to you. We have vitamin D, progesterone, estradiol, which eventually converts to estrogen, and aldosterone, thyroid hormones, and very important nuclear receptors in the body. Now, I want to talk a little about nuclear receptor superfamily. It's the largest nuclear receptor superfamily, hence the term superfamily. But we have this first category for nuclear hormone receptors. These have their matches. So the ligand has the nuclear receptor that it works with. Now, you have this category, which is the orphan category. These do not have their matches all the way. So you have like fatty acids, phospholipids. It's still questionable what the ligand will bind to. Now, this category is the adopted orphans. And these two highlighted in blue is what Dr. Schrodinger spent a lot of his career on, primarily the pregnane X receptor and its role with xenobiotics. And that's what we focused in the lab this summer with my project. So you might be asking yourself, why is this important? Now, it's important because herbal supplements have a lot of unknown effects. Now, and you put these with other drugs or medications, it can have unintended health consequences, and they could lead to negative outcomes with the patient. The most famous example is St. John's wort. St. John's wort is a supplement used for depression and to boost mood, but it induces the cytochrome P450-3A4 enzyme, which is a major enzyme in the liver and intestines, and it metabolizes drugs, but it metabolizes a lot of drugs. And in research, it's been shown that when you're taking it with medications such as oral contraceptives, it can lead to pregnancy, which means that there is a drug-to-drug interaction, making the oral contraceptives less effective. Which leads us to our hypothesis for this project. Herbal supplements may activate PXR, affecting its target gene expression, changing the pharmacokinetics of other treatments. So to do this, we started out with, we tested 6 extracts. So we tested neem, kratom, lion's mane mushroom, cordyceps mushroom, and we tested black licorice and black cumin. So we did this first step, plasmid amplification and isolation. So we took DNA fragments, we amplified them, and we purified them for use later on. Then we took our extracts, we made extracts. So we took the supplements, we made it into an ethanol extract and a water extract, which we later combined together. Then we did a luciferase reporter gene assay. So we used the luciferase to show the presence, if there was presence of PXR, because it makes a bright light in the presence of PXR. Then after that, we took some LS180 cells, which is a type of colon cancer cells, and we treated them with the drugs to see how it would react. And after that, we isolated the RNA and put it into the qPCR, which is quantitative polymerase chain reaction, for analysis. Let's go over some results. So with the luciferase, on this first plate here, we have the black licorice and black cumin. Now we did this in a dose-dependent manner. So we started with the low dose and ended with the high dose. We didn't find the threshold dose in our experiment this time, but you can see that there is some higher fold induction, meaning that there is activation. Now on the second plate, we had kratom, and we also had lion's mane mushroom. But when we went to do the reporter gene assay, the lion's mane didn't get enough of the solution, so we were not able to get results to produce in a graph. So we just have kratom, but this does show a higher induction with activation as well. Now on this third plate, we have neem and the cordyceps, which is another type of mushroom, but we also have our positive control rifampicin. Now rifampicin is an antibiotic, but we know it already activates PXR, so that's why we used it as the control. So let's talk about the qPCR results. So we tested it with other nuclear receptors such as the CYP1A1, the CYP7A1, MDR1, PEP-CK, and the SHP. Now this graph does show that kratom and black licorice as well as neem, all induced the CYP1A1 gene, or the nuclear receptor. But black licorice also induced the CYP7A1. So we had some limitations with this project. The first one being that when it comes to the reporter gene assay, it really forces overexpression of these nuclear receptors. Now, this can lead to false positives sometimes when it comes to these results. And like I mentioned earlier, when we did the lion's mane plate, it didn't get enough of the solution, so we were not able to show if it did activate PXR or if it was toxic to the cells at the time. And since we used those colon cancer cells, we grew them in tissue culture, but that's only looking at one small part of the body. That's not representative of how the whole body would react or if it would activate PXR in other parts of the body. And then with the solutions we used, they're crude mixtures, the extracts we made. So what did we learn? We concluded that black licorice root, black cumin, neem, and kratom, they all activated PXR in the reporter gene assay through our dose-dependent manner. As the graph showed, the higher doses showed more activation than the lower doses, but like I said, we didn't find that threshold dose, so higher doses could show more activation. And then once we treated those cells, black licorice was the only supplement that induced the CYP3A4 gene. But black licorice also induced the CYP1A1 and the CYP7A1. But kratom and neem also induced the CYP1A1. So what would it look like in the future? I think there's a lot of directions that can go with herbal supplements and testing. It's vastly unexplored, but the main focus, I think, would be to see how it reacts in the inflammatory pathways. Lion's mane and cordyceps and other mushrooms, research says that it should have anti-inflammatory properties or activity, but it hasn't been confirmed all that well. I think there could be more testing done to see how those mechanisms work. And then we could treat colon cancer cells or different types of cancer cell lines with these supplements to see if they induce inflammation or not. I would like to thank the MSSU KC Research Consortium, Dr. Kramer, Dr. Staudinger, Dr. Gordon, Dan and Catherine, the NIH grant, and the Provost Office for supporting our student research. That's my references. Any questions?
I'll start a question for you, Whitney. So great job and really impressive. You know, one of the differences that we'll start seeing now with our dental students who are transitioning from year 1 to year 2, joining the SSRF, is that this was all done in 4 weeks instead of 8. So that's really impressive. Thank you for the hard work. So transition, or tell me about what you learned from this that may be beneficial as you become a practicing dentist.
Yeah, I took a lot away from this experience. For one, it helped me kind of put together all the concepts we learned through our biomedical clinical sciences. You learn you see the PowerPoints, you learn specific aspects of mechanisms and whatnot, but seeing how it reacts in real cells and like how it reacts in the real body kind of helped put together how I would look at it in a patient, how you would look at not just the chief complaint they're having, but the rest of the person as a whole, and see how I could treat them, you know, help them with other symptoms. I now understand like cancer patients, kind of what they're going through, what their body's going through, and how it can affect them in a better way.
First of all, congratulations. As Dr. Kramer said, you have done this in 4 weeks. That's very impressive, in addition to the curriculum. I want to congratulate you also on picking a topic that's really important. As you said, we know very little about herbal supplements, and yet lots of people take them and believe different things about them. So I really applaud your interest in building the evidence base around herbal supplements. What was interesting is what kind of impedes inflammation versus what doesn't. And I'm thinking, have you thought about diseases that are caused by inflammation, like IBS, where you know there's lots of gut inflammation. Is there any research out there? Did you come across anything where any of these herbal supplements inflammation may be helpful to individuals who have these inflammatory diseases?
Yeah, I think that's a great concept. There isn't a lot of research with herbal supplements and inflammation. That's why I know a couple of my other colleagues kind of dove into that a little bit throughout this summer to see what that would look like. But I think there's a lot of room to look at inflammation and how, if it could help or not. Time? Okay.
Thank you.